Well, thanks for having me here today. I'm going to start today by introducing you to Tommy Francis Jr. And many of you probably know of Tommy. He is the author of this famous essay on the doctrine of original antigenic sin, which was published in 1960. Everything that my lab has been studying and everything I know about human immune responses to viruses are consistent with the theories that Tommy put forward 60 years ago. And he's just a remarkable scientist to think about what he was able to accomplish with some red blood cells and some virus isolates. So I'm going to come back to Tommy at the end, but I want to acknowledge him and the long impact that he's had in thinking about how humans who are exposed to diverse viral strains over the course of our lifetime, how we respond to new viruses as they emerge. And what Tommy figured out, and what is certainly true in the context of influenza virus and probably many other pathogens, is that there is something magical about childhood. These are my two kids several years ago. They're older now, 7 and 9 now. But what do we do with our kids? We read them books. We let them get messy with paint. Why on earth would we do stuff like this? Well, we read them books. Not because they're going to remember those books 30 years later. We read books to, a 1-year-old or a 2-year-old because studies have shown that if kids have books read to them when they're very young, they will go on to become good readers. It's almost as if those words going into our little kids' brains, early in childhood, it's almost as if that programs those kids or imprints those kids in a way to understand language as they develop as adults. Well, guess what? It's not just things like paint and books, but what we're learning, again, based on Tommy Francis's principles long ago, is that viral encounters in childhood have the same long-lasting effects. And in fact, what we're learning is that our exposures with viruses as we are children set up lifelong memory responses that we draw from the rest of our life. Now, my lab focuses primarily on influenza viruses, and in the case of flu, we are exposed to flu viruses by the time we are 5 years of age. This is a nice study demonstrating that. It's a seroprevalence study from the Netherlands where childhood vaccinations are very rare. You can see if seroprevalence to H1 and H3, the two influenza A subtypes that circulate in humans. Some of us have detectable levels of antibodies upon being born. These are maternal antibodies. These wane over time. And look what happens. By the time we're about 4 or 5 years of age, essentially all of us have encountered influenza. Now, what happens during those initial influenza encounters? You get the induction of these long-lived memory B cells. The beauty of our adaptive immune system is that we have zillions of B cells and T cells, right? You could throw any antigen at me today. I'll probably have a few cells that can respond. Now, in the case of flu, we get infected by the time we're 3, 4 years of age. And these naive B cells proliferate. They undergo something called class switching, affinity maturation. And over time on the x-axis here, you get what we call memory B cell pool. Now, there's two important things about these memory B cells. Number one, there's more of these guys compared to before the initial viral encounter. But what's perhaps more important in the context of flu is that these memory B cells are at not only a higher level, but they have a lower threshold of activation. Okay, you just tickle these guys a little bit and they take off. In the context of influenza, we're exposed with viruses when we're 3, 4 years of age. I can now be exposed, 30, 40 years later. Those B cells can get recalled from those initial childhood imprints, even if they're mismatched to these antigenically drifted strains that I encounter today. And again, it's this crazy competition between memory cells and naive cells. But in the case of flu, memory cells usually dominate the response, not because the BCRs are the best match. They often are not, because these viruses change so much over time. But they end up nonetheless dominating the response because of this low threshold of activation. I think that's something that people don't pay enough attention to. I will talk about coronavirus soon, but I have at least one more slide on flu. Now, when we think about flu over the last 100 years, there have been many different flu subtypes that have circulated. This is something my lab and others have studied quite a bit over the past few years, that what we've discovered is that, say you're like a super old person that was born in 1968, a little nod to the 50-year-old plus crowd, If you were first exposed to flu in 1968 with one of these viruses called an H3N2 virus, we've learned that you have a fundamentally different immune response compared to, say, someone that was much younger than you, born in 1978, who was exposed perhaps to one of these H1 viruses. Okay, and studies have shown, for example, there's a study, Gostick et al. From Mike Warby's group that I love, which has shown that H5 and H7 pandemic susceptibility, these are different influenza subtypes that cause limited human infections, but we worry about as far as our next flu pandemic. Those studies have shown that if an individual was exposed to H3N2 virus compared to H1N1 virus in childhood, that they actually have different susceptibilities to these new pandemic viruses. And that's because there's some shared epitopes in what we call the hemagglutinin stalk domain. And it's even more complicated than that. So, for example, my group has shown that if an individual is exposed to an H1 virus in 1977, that they have a different antibody specificity to today's contemporary strains compared to someone that was exposed to an H1 virus in 1985. It really is like pretty crazy to think a 6-year birth difference, you have a whole different set of viruses to prime your immune system. And that's because not only do, you can see over this timeline, they have viruses been in the population at different times, but they're constantly changing. So a 1977-78 flavor of H1 is different as far as something to get your immune system going compared to a 1985 flavor of H1. So we've been studying this again in the context of flu for a long time, and so we know that immune history, particularly these childhood infections, influence the specificity of flu antibody responses, and this we have nice demonstrations on how this affects susceptibility. And so it was a natural thing for our lab to ask the question, could something similar be going on with coronaviruses? And to me, when I think about this question, there are 3 striking things about SARS-CoV-2. Number one, there's a whole lot of heterogeneity in disease outcome. Okay, I can get infected, my wife can get infected, I might die, and she might be completely asymptomatic. I mean, that's insane, right? There's so much different outcomes following infection. The same is true with immune responses. And this is a study that we did with John Weary and our group at Penn early in the pandemic, showing that, individuals with different degrees of disease have super high variability, for example, in IgM and IgG against the SARS-CoV-2 spike protein. These people are all severely, these are ICU people. You have some people that have no antibody response at all, other people with screaming high antibody responses. Heterogeneity in disease, heterogeneity in immune responses. And here's another one, here's the third one I want to talk about, is this age-related, or perhaps year of birth related, difference or mortality. You can see old people here, they tend, and this is, everyone here probably knows this by now, right? Old people tend to do far worse compared to younger people when it comes to SARS-CoV-2 infection. Now, I have age-related or year of birth related in parentheses, and I think that's something to separate, okay? So, for example, I'm born in 1978, and I have a certain age right now, but in 10 years from now, I'm gonna be 10 years older, but I'll still be born in 1978. And us flu people really think about this year of birth thing because of that timeline I showed you with influenza circulation, because it really is not so much an age thing with flu, it is more about what kind of viruses you saw in childhood. Now, in case of SARS-CoV-2, I think I hypothesize it's both, right? Clearly a deteriorating immune system over time is not a good thing. But those questions remain unanswered. How much of this disease curve as it relates to age, how much is that about the kind of viruses we've been primed with in childhood compared to our bodies falling apart? Okay, so if we think about coronaviruses and what have circulated over the past, well, SARS-CoV-2 is, as you've heard today, is a beta coronavirus, okay? There are at least 2 major alpha coronaviruses, NL63 and 229E, which are common coronaviruses that have circulated in humans for quite some time. And there are 2 beta coronaviruses, OC43 and HKU1, which are human common beta coronaviruses that have circulated for some time. Now, I will just say, as we think about that slide with flu nicely laid out of what circulated over the last 100 years, I'll tell you, these estimates are a little dicey, okay? And and I think it actually matters if you know, how long have beta coronaviruses been in the population? Is it really 120 years, or is it more like 50 or 60 years? I will say that I think we're going to learn more about that as people refine these estimates of when these viruses might emerge. But one hypothesis is that if you're infected with an alpha coronavirus, for example, as your first exposure, that you'll have a different type of immune response to SARS-CoV-2 compared to someone that was first introduced to coronavirus in the context of a beta coronavirus infection. And how that relates to year of birth, or is it just sort of stochastic, whatever you end up with, it's not really clear. These are, I'm going to give you more questions than answers today. Okay, so there's been a lot of SARS-CoV-2 papers over the last couple of years, over the last year and a half. This one really has caught my attention. So this is a JCI paper that looked at electronic health records of people that were infected with SARS-CoV-2. And they looked through the records and they just simply asked, well, how many of these people had a documentation of a common coronavirus infection previously? And they sort of stratified that, right? People that either did or didn't. And then they looked at disease severity. And what they found are that people that had a recent common coronavirus infection had less illness upon SARS-CoV-2 infection. Okay, so this study suggests that these common coronavirus infections, at least if you had one recently, that it didn't prevent against SARS-CoV-2 infection, but it did limit disease upon infection. Okay, I'm going to come back to this in a minute. We started our studies just like we would with our flu studies, where to look at this question of cross-reactivity of common coronaviruses and SARS-CoV-2, we started by looking at a large panel of serum samples collected several years before the pandemic, in this case, samples collected in 2017. And we analyzed samples from individuals with a diverse range of different birth years. And we did especially high sampling of kids in this particular study. Now, in this first experiment, I want to show you, this is just an ELISA where we are measuring antibodies to these two common alpha coronaviruses and OC43, a common beta coronavirus. We didn't use HKU1 because when we were doing this back in the day, you know, months and months ago, we just simply didn't have that protein yet. What you can see is that this is against the spike protein. You can see that most of us are exposed, just like flu, we're exposed to these both alpha and beta coronaviruses by the time we're 5, 6, 7 years of age. These samples were collected in 2017. So you can see by the time we get to, at this point, a 7-year-old, 2010, just about everyone is seropositive. Okay, so these viruses are plentiful. It's hard to, you know, walk around in, these are all from Philadelphia. It's hard to walk around in Philadelphia as an infant and not get exposed to these viruses. And so we wanted to investigate the possibility that perhaps these viruses are eliciting some sort of cross-reactive antibodies against SARS-CoV-2. And so we actually, as we set these experiments up, we didn't really have dreams of you know, publishing papers at this point. We were doing these experiments as we were trying to develop serological assays in those first weeks of the pandemic. But what we did was we took these pre-pandemic samples, and then we got samples from people that had just recovered from SARS-CoV-2 infection, and we tested these samples against the full-length spike protein of SARS-CoV-2, receptor binding domain of the spike, or the N protein. And what you can see is that, of course, with people that are in red, these people are convalescent. They have antibodies against all three antigens. But there is some appreciable cross-reactivity. 4% of people in 2017 in Philadelphia had antibodies against the full-length spike. Fewer people had antibodies against the receptor binding domain. Receptor binding domain, as you heard in other talks, is a smaller region of the spike. That's why this is your antigen of choice for seroprevalence studies. There's low, pre-pandemic cross-reactivity. Look at the N protein, right? 16% of people had antibodies against the N protein, and this makes sense because the N protein is much more conserved with common coronaviruses compared to the spike protein. We looked at year of birth. I will tell you there was nothing as far as year of birth. I don't think the case is closed on this. I'll talk about this more in a minute, but when we stratified these data, looking at cross-reactivity of pre-pandemic samples by year of birth, nothing really shook out. You can see the trend line here and individual samples in the circles. Very low frequencies, but real frequencies of antibodies against the spike. When we look at the N protein, the same kind of deal. It might look like there's more dots here, but there's just more pediatric samples in general in this collection. So I don't think this means that there's not a year of birth effect. Again, I'll touch on this later, but when we looked at these samples that had cross-reactivity, they were at similar levels in all the age groups we looked at. Now, it's a big question. So what do these antibodies do, right? We're all exposed to these common coronaviruses. Some of these antibodies elicited by common coronavirus infections cross-react to SARS-CoV-2. Do they protect us? Well, first we did neutralization assays. In this case, this is a VSV pseudotype neutralization assay performed by Paul Bates. We've also done, Sarah Cherry has done BSL-3 bona fide neutralization assays. Same color scheme here. These are the individuals that had recovered from SARS-CoV-2 infection. You can see most of them have neutralizing antibodies. And regardless of whether or not someone tested positive for cross-reactive SARS-CoV-2 pre-pandemic antibody, we found very low levels of neutralizing antibodies. And this is more or less just background in this assay. So, okay, so that's in vitro. What about in vivo? And it's hard to obtain those samples, right? Because how would you ever obtain samples before a pandemic and be able to link that to whether or not someone was infected? Well, we have the Penn Medicine Biobank, which I like to joke, if you go into one of our hospitals, just to use the restroom, there will be a nurse that like runs up to you and tries to bleed you, because we are routinely trying to consent people coming to the hospital for a variety of reasons, right? Just routine surgeries, things that are more serious. It's actually an incredible resource, the Penn Medicine Biobank. They have a slogan: a few minutes of your time and a simple blood draw can impact the future of medicine. And isn't that true for this study? Because there's thousands of samples collected, hundreds or thousands a day, probably. It's insane. But we were able to identify 251 individuals who donated blood before the pandemic, who went on to become PCR confirmed infected with SARS-CoV-2. And we compared antibodies in these individuals compared to an equal number of individuals who just didn't get infected. And this included samples from 80 people that were actually hospitalized. So we were able to look somewhat at disease severity. Okay, so first shown here are antibodies against the full-length spike in the middle here, I guess, the receptor binding domain, the N protein, and here's against OC43, one of those common coronaviruses. You can see that antibodies against the receptor binding domain are super rare. Nothing, you can't really tell anything there. Here's one person that got infected that had very high levels pre-pandemic. Against the full-length spike, again, there's just a smattering of samples. There's nothing statistically significant between the groups. Same thing with the N antibodies and the antibodies against the full-length spike OC43. So nothing really looked, protective or correlating with protection. It's known that antibody levels against common coronaviruses decline over time after exposure. So we hypothesized that we weren't seeing an effect here because these samples were collected over multiple years. In this case, we looked at the samples within this cohort that were just collected within the previous 12 months to see if we could see a signal there. Nothing, right? So the receptor binding domain spike antibodies are rare. In this case, no one even had them. The spike, again, pretty rare, especially when you look at this abbreviated group, and no difference in N antibody or OC43 antibody comparing those that got infected and those that were not infected. We also looked at hospitalization, the need for oxygen, these kind of things. Nothing shook out. We saw no correlations with protection. Okay, so it's interesting, right? Does this, is this, I try to talk about heterogeneity of disease. It seems like some of us have these cross-reactive antibodies. We don't understand why, right? Like, we're all exposed to these common coronaviruses. Only some of us actually have these cross-reactive antibodies, but they don't seem to prevent infection. And at least in this hospitalization versus non-hospitalization cohort, we didn't see any effect. We next wanted, I'm going to get back to this whole correlative protection in a minute, but let me just complete, the thoughts on this sort of, as we go through an infection here. So what actually happens upon infection to these antibodies, right? So they don't really prevent you from infection. Well, what happens, do they help you recover? In this case, we looked at this cohort. This is a cohort at Penn that included healthy donors, recovered donors, and these are people in our ICU. So we published a study early in the pandemic on this cohort, but here we completed in-depth antigenic analyses with 27 of these severely ill individuals. And we measured antibodies against these alpha coronavirus spikes, as well as a common coronavirus, beta coronavirus, and SARS-CoV-2. And day 0 is time of hospitalization. Day 7 is 7 days later. You can see upon hospitalization, as I showed you in one of those earlier slides, people had varying amounts of antibodies upon, when they actually walked in the hospital. You can see that this got boosted over time. And look at alpha coronavirus antibody titers, no difference, right? They level off. But what you see is in some people, you have a boost in these OC43 antibodies. And you could express this as fold change. You can see, of course, people that are undergoing a SARS-CoV-2 infection have cross-reactive or have antibodies boosted against SARS-CoV-2, but you also have these cross-reactive antibodies against OC43. And in some really neat studies that we've just done or unpublished, some of these antibodies paradoxically cannot even react to that boosting antigen. Remember what I said about those memory B cells having a low threshold of activation. So where are these antibodies against? Well, they're against the S2. That's where the most homology is. And so if you look, this is the same patients. This is OC43 full length S1 or S2. You can see you don't get a boost against S1. All those antibodies are reacted to the S2, which are more conserved between OC43 and SARS-CoV-2. We don't see any difference in disease. So this is looking at the extremes, who survived and who died. You can see pretty much across the board, we see these increases in what flu people would call a back-boosted antibody response to OC43. Okay, so I want to return to this, and I probably don't have much time. I probably have like 5 minutes. So what is going on with this finding, okay? Because I believe this study, right? This is a well-done study showing that people that had recent infections actually have less disease. I want to present here another piece of the puzzle. I just received an email 5 minutes ago showing that this paper is actually accepted. So it's on medRxiv, but it'll come out soon, I guess. In this study, we set up a cohort of healthcare workers in Philadelphia right from the start of the pandemic. And we obtained blood samples from them over the course of this entire summer, okay? And just like the rest of the US, we had a huge spike of cases in Philadelphia, SARS-CoV-2 cases in Philadelphia over this fall and winter. And this was nice because we had these samples from people that we knew had never been infected with SARS-CoV-2 because we've tracked them a lot over the spring and summer. And then we were able to see how they reacted upon SARS-CoV-2 infection. Okay, so let me walk you through the data. I think this is very interesting. Okay, so consistent with what I just showed you in that paper that we published in Cell earlier, I guess, I don't know, and I think it was in 2020, you could see that in this cohort, there was no RBD antibodies. When we looked at full-length antibodies in people that were not infected or infected, these are healthcare workers, again, where we have blood samples before this second wave. You can see there's variable levels of antibodies against the full length. There's variable levels against the N protein, but nothing correlated with protection. There's nothing there, right? I mean, they're pretty rare and they're equal in both groups. Now, this is where it starts getting interesting. When we look at antibodies against these seasonal coronaviruses, in this case, now we have HKU1 in here as well, that there are equal levels of antibodies against the alpha and beta coronaviruses in the healthcare workers that either did or didn't get infected.
Scott, this is a 2-minute warning.
2-minute warning. All right. So here's the cool thing. Now since we had these samples that were collected right before these healthcare workers were exposed, we then gave them a survey and we said, what were your symptoms like? This is where it gets really cool. So if we look at this, if we look at the people that have the rare cross-reactive antibodies to the SARS-CoV-2 antigens, nothing really shakes out. These were very infrequent. Now look at this. In white and yellow are people with more mild illness. Their disease is either no symptoms or symptoms resolving within 7 days, symptoms resolving within a month, symptoms longer than a month. You have a clear trend with people that either didn't have symptoms or symptoms that quickly resolve. They have higher common beta coronavirus antibody levels compared to people that had longer durations of symptoms. And this is specific against beta coronaviruses because when we do the same thing with alpha coronaviruses, we don't see it. Okay, so here's the question I will end with today. How can pre-pandemic common coronavirus antibodies, but not SARS-CoV-2 reactive antibodies, limit symptom duration following SARS-CoV-2 infection? That doesn't make sense, right? Because I showed you it's the common coronavirus antibodies. If that was antibody mediated, then you would see the same correlation with the SARS-CoV-2, pre-pandemic reactive antibodies, but we don't see that. And I turned your attention to this Nature Medicine paper from 2020. This is go take a look at this paper. But the authors of this study monitored serum levels of common coronavirus antibodies over time in a well-documented cohort, and they have documented infection. So here, this is looking at antibodies to OC43. Here, this person got infected with common coronavirus. You can see they have an antibody response. It goes down over time. Here, they got reinfected. Antibody response goes up, comes down. In this way, common coronavirus antibodies are simply an immunological stamp that marks how recently you were infected with common coronavirus. Okay, so I don't think that these antibodies actually protect. They're not neutralizing. They recognize parts of the virus that are unimportant, but they are an immunological marker of recent infections. And that is consistent with that JCI study that I just laid out to you, that if you're more recently infected with a common coronavirus, it seems that you have protection, not against infection that's not neutralizing, but protection against severe disease. And given the fact that we don't see the same correlation with the cross-reactive SARS-CoV-2 antibodies, I bet you this is at the level of T cell immunity, or perhaps some memory B cells that are just not at a high enough frequency and differentiated so we can see the antibody. All right, I'm probably out of time. So let's just end with this. Let's end with Tommy Francis. So Tommy Francis, could have predicted all this stuff 60 years ago. I think there's still so much to learn. I don't think the data I showed you today shows that there's not a birth-related effect. We don't see that in the serological component, but that's the same thing with flu. We only see these responses upon a boost. And so our lab's very interested in looking at the cellular response, both B cells as well as T cells, to see how different childhood exposures might shape our immune responses to coronaviruses, how this relates to pandemic susceptibility. And this idea of recent exposures is a big one. We're very interested in studying this to see if this might explain some of the heterogeneity that we see in the population, particularly kids that are exposed to these viruses all the time. Does this relate to why we have protection or very mild disease in kids? Thank you for having me today. Hopefully we have time for some questions, Eric.
Awesome. Thanks, Scott. I do have a question I'd like to lead off with. And again, I remind attendees, please raise your hands and I'll call on you in the order that I see your hand. So if I understand correctly, we have all been exposed to either an alpha or a beta, or perhaps both coronaviruses sometime early in childhood. Is that correct? But then roughly 16% of the population shows some kind of antibodies against the N protein, right? With respect to the patients that would be categorized as recently infected with a beta coronavirus, what fraction of the total US population falls into that category? Just ballpark if you don't know the number.
Yeah, I wish I knew that number, Eric. And, I'm sorry, I forgot this slide, which is the most important. And I'd like to thank all the folks in my lab, especially Sigrid and Liz. And you can see this is just a huge team effort. Folks at Penn have been great. I get funding from boring people like the NIH, as well as cool people like Joel Beed. I've been trying to work with modelers on this question because, I want to know, for example, Eric, does this person here with a titer of, these are arbitrary units, right? Someone has a 32 microgram titer. On average, how much time has gone by since that person saw OC43 compared to this person with a 2 microgram titer? And it seems like I don't know anything about computers, but modelers and really smart people should be able to figure that out based on these kind of studies where, of course, there's a lot of human variation, but this study is really nice. They track, a decent sized cohort over time where you should be able to look at these decay ratios and on average ballpark what a difference of antibody titer equals to a days since last infection. I'll be honest, I have no idea if someone like this versus this, if their exposures are 5 years different, is that on average, like a 5-year difference in exposure, or is that like a 6-month difference in exposure? This isn't quite getting at your question, but the answer is I don't really know how many people on a given year are exposed to common coronaviruses. And I don't know how our antibody titers relate to the timing of infection. But these are things we want to learn. It's possible that we're always exposed to coronaviruses, and every now and then one catches, and then you get a flaming infection, and you get a real big boost in your antibody response. These are things that we just don't know. Not to hand wave, but it really is the unknown.
And within the data set that you were just showing in that slide, that was collected, if I caught it correctly, from UPenn medical staff. Is that right?
Yeah, so this is this paper is online, and I can send you the link for this. But these are healthcare workers that have direct patient care. We enrolled people in March and April as the pandemic was starting. We collected blood samples every 2 weeks until the summer. And then at that point, and we monitored for NP swabs and whatever during that first period. And then we stopped collecting samples. But then what we did was we monitored how many people, where we already had these summer samples collected, how many people got infected during this second wave of SARS-CoV-2 activity in Philadelphia. So these are healthcare workers. Our previous studies have shown that their seroprevalence rate is lower than the rest of the population. The demographics are lower. They might have unique coronavirus exposures, right? Maybe just working in the hospital over the past years have given them unique coronavirus exposures.
Perfect. And I guess the question I was driving, and I think you answered it, is the way you've designed the study, you would not have overlooked asymptomatic individuals. So they're fully represented in the study that you did, right? Because you're monitoring everybody.
It's a great question. So we do have people that are completely asymptomatic who are the white dots here.
Yeah.
There's only a handful. During this first wave of the study, we were monitoring these people much more carefully. During the second phase of the study, we gave a survey out in January, and this is self-reported.
Yeah, okay.
So if someone was asymptomatic, then they got tested for one reason or the other, right? It's not like these healthcare workers were getting NP swabs, every 2 weeks or something.
Oh, so you would have missed people who weren't getting tested because they were asymptomatic, or?
Yeah, that's fair to say. There are probably some asymptomatic people that we didn't capture here.
All right. We have a question from Ina Ricardo Lax. Ina, you can unmute yourself and ask away.
Thank you. I have a question about the previous slides where you see kind of the reverse cross-reactivity between SARS-CoV-2 infected people who now have some cross-reactivity with OC43.
Yeah.
I was wondering if you saw the same thing in individuals that have been vaccinated.
Very good question. So we are we're putting together a paper on this right now, and it is super interesting. Here you can see that there is pretty much across the board these boosts in some people upon SARS-CoV-2 infection, in some cases, tenfold increases in an OC43 boost. Now we have assays up in our lab now where we do absorption type of assays where we coat, for example, SARS-CoV-2 spike onto a bead. We add this to these serum samples, and then we pull down whatever sticks, and then we redo the ELISAs. This is a way for us to look at cross-reactivity. Tommy Francis did this with flu viruses with red blood cells. He would actually put virus on red blood cells and use them as the absorbing antigen when he did this with his flu studies. What's really interesting is that these OC43 antibodies in these assays that I just described to you, they get boosted, but they don't bind to SARS-CoV-2 spike in these absorption assays. I mean, they have to have some sort of reactivity, right? Because SARS-CoV-2, stimulated the B cell receptor. What we think is that these B cell receptors, when they're expressed, there's 10,000 B cell receptors on a B cell. So the affinity is so low on an individual antibody level. But when you put 10,000 of them together, there's enough avidity to get a memory B cell to start secreting these antibodies. Now, the reason I just walked you through all this, I know you asked about vaccination, because now when we look at vaccination with mRNA vaccination, you see some boost in antibody. It's nowhere close to what you see during infection. You see a little bit of a boost of an OC43 response, twofold in some people, threefold. Here's the cool thing. Those antibodies are totally cross-reactive to SARS-CoV-2. When we do that same absorption assay, SARS-CoV-2 can pull down all of the antibodies that are OC43 reactive that are boosted. So SARS-CoV-2 mRNA, the mRNA vaccines deliver antigen in different ways compared to infection. We've shown in other studies, we do a lot of flu work with mRNAs, they elicit these strong germinal center reactions. There's almost like this persistent antigen expression, which we think tilts more towards a de novo response. You have a lot of antigen, you have germinal centers that are set up, and you get these de novo specificities. You get some of those C43 cross-reactives, but they're just legitimately like these S2 antibodies that are just cross-reactive. Whereas infection, it's sort of just like the flu story. You're at the mercy of your memory, and you start pulling out some of these antibodies that are just not that useful because they don't stick very well when they're secreted as an antibody.
Okay, gotcha. Thank you.
Okay, we have a question from John Hunt. John, you should be able to unmute yourself and ask your question.
Yes, interesting talk. Thanks. Thinking ahead to the booster vaccine issue, you've shown that older infections from other coronaviruses don't do much. I would assume that older infection from this same virus would be better. Do you have a way to estimate from data how long protection is likely to last, assuming there isn't some significant antigenic or virulence shift?
Infection from or protection from like
from reinfection, sorry, from either a natural infection or vaccine.
For the SARS-CoV-2, you know, if you would have asked me a year ago if people that were infected and recovered would need a vaccine, I would say, why would you do that, right? But the mRNA vaccines elicit such a high level of antibodies relative to infection. And, you know, if you look at people that are recovering convalescent from infection and you test their antibodies, for example, like what Mike was showing, Mike Diamond, and for some of those drift things, we see the same data, right? You get this 8-fold reduction. If you look at infection-induced antibody responses, you start out at such a low level relatively. There's a lot of heterogeneity, and the average is like, you know, pretty low. And so when you get that 8-fold reduction, those infection-induced antibodies hardly, you know, in some cases, I think you probably have these breakthrough infections. Vaccination, on the other hand, you start out at such a higher level. And if you're infected and then vaccinated, a bunch of groups have shown this, including John Weary from our university. If you have infection and then a single vaccination, now you get those very high levels of antibodies. So the levels of antibodies are important, I think, you know, when we start thinking about these drift variants. And then the longevity, we have done a study with flu mRNA vaccines where we immunized mice. This was published in 2017. And I didn't know that my technician kept them around, but she kept them around for like 400 days. And the antibody response in those mice 400 days after vaccination was the same as 21 days. There, you know, it's mice compared to humans. I don't think we have to worry. I just got my booster 5 days ago, and I feel like maybe we'll have to get boosted with like the South African strain, but I don't think so at this point. We'll have to see. We'll have to monitor antibody levels over time, but I would expect that these vaccines are going to elicit high levels of antibodies that can recognize drifted forms, and that, you know, if you get the mRNA vaccine prime boost, you're good to go. Other vaccines, I'm not so sure about. You know, you look at, you know, inactivated coronavirus vaccine in China, it's hard to get data from that. We're going to start a study with Hong Kong, and we've already been told that I don't know if we're going to actually do it, because if we go on to show that the inactivated vaccine is far, you know, inferior, then, you know, I'm not even sure. I think the government will be mad at us. So it's like, that's like a lose-lose situation doing that kind of experiment. But, you know, I think those kind of studies need to be completed where we compare different vaccines. mRNA vaccines, whatever. I'm at Penn, and like Penn has, you know, all, you know, obviously tied up with patents and all this stuff. But I really think that, you know, an unbiased person can look at these vaccines and realize that they have the chance to really just change vaccinology, because they elicit such high levels of persistent antibodies. So that was a very long answer to your question, but I think
Yeah, but thanks. I got it.
Even if you're infected.